Hello and welcome everyone at MayaSat Biosample Overview. My name is Bence Mátyás. I'm the Science Director of the Genoplant Research Institute. We are a Slovenian startup and we are advancing microgravity research by building smaller payloads to low Earth orbit and beyond. And now our debut mission, the MayaSat-1, is going to launch very soon from Vandenberg. Only 1 month left in June, targeting a June launch from Vandenberg, California for a polar orbit. And the good thing is we are also going to retrieve all of these biosamples. Many of them were not flown in space before, and we are going to showcase them, our customer payload, our own research, and the variety of samples which are going to be available for a wider audience who want to support and contribute to open space seed database. So the house rules, please keep your microphone muted until we are not reaching the Q&A question or until you are not invited to the stage. You can drop your questions or comments anytime to the chat box. You can find down there the icon, and we are going to read your comments and questions at the Q&A part. This event is going to be recorded, and we are going to share with our communities and with the people who missed this event, basically. A brief introduction about my background, just in one line. I'm coming from the environmental sector, from the soil microbiology. And basically with the Genoplant, our main focus and our core product is microchip electrophoresis. It's basically separation science. So we are detecting different ions and molecules based on their charge and size in different liquids, basically soil and plant samples. And with that, we are learning many things about their stress responses. At the today meeting, we are going to have an overview of the MayaSat-1 and the mission profile, the mission possible profile 2025. We are briefly going to introduce our customer payload and the available research payload, what is going to come back to Earth. The next steps, and you can also address your questions at the end of the meeting. So the MayaSat-1 is a biological incubator, which is carrying passive payloads, hundreds of different biosamples. And it's a one-unit CubeSat, but it's not deployable, the first version, because the MayaSat is integrated to the Mission Possible space capsule in a pressurized compartment. So because of them, we're able to also retrieve the samples, not only launching to orbit. And the second version of our CubeSat, we are going to be deployable. Every mission has its benefits and disadvantages. The MayaSat-1 can be retrievable, but it cannot be deployed to its own orbit. So here is a rough overview about the biosamples, what we are having. So the biggest area, which we cannot read it here because of the tab, but this is the seeds, plant seeds. And we have algae on board. We have fungal biofilm, from which we are going to hear more about today. We also have a soil microbial experiment, and we have plant tissue, and we have human DNA studies also onboarded MayaSat. MayaSat-1 was ready in 2024 September and handed over to the exploration company in Germany, Planegg, in October 25, 2024. So from Slovenia, we basically drive and transfer our CubeSat, and we had an opportunity to have a company visit, and we could see the Mission Possible in the making. So that was a very great opportunity to see actually the spacecraft which is carrying our payload. This is exactly where MayaSat-1 is located. This is part of the pressurized compartment. Later you are going to see more photos about this. So basically in the pressurized compartment, the MayaSat-1 and among other payloads are being placed in the bottom part, and on them, the larger satellites from ESA, I believe, and other bigger companies and space agencies, their payload is placed here. And that's how you can imagine in size compared to humans. It's the mid-size demonstrator version of the NIX space capsule. in the Mission Possible version carrying 300 kilogram payload. And this is the recent post from the exploration company. So they shared an update that the capsule is being sealed now to the USA, where it's going to be integrated with the Transporter 14 mission. So it's going to be very soon, and we are very happy to be on board in their capsule. So we are going to receive soon the official launch invitation that we are going to forward you. It's supposed to happen 4 weeks before the launch date. The launch date is expected in the second half of June. So if you want to go to Vandenberg, there is going to be a launch ceremony for our customers, going to be represented by the Orion team, by Anette Fenyőfalvi, and she's organizing there near to the launch pad an event. So if you want to see the launch on site, it's a very great opportunity. Otherwise, the other part of the team who cannot go to US this summer, we are going to celebrate in Germany because the exploration company invited us. In Planegg, they are going to organize a launch ceremony. If you are interested to participate in any of these events, please let us know. And so it's a Falcon 9, the launch vehicle, and it's a very special launch site that was one of the reasons that we decided to go with the exploration company, which we are going to share in details. Among the payloads, the MayaSat-1, which we are very proud of, designed and made in Slovenia, and also their Australian project, the Rescue, and also other customers who could catch a ride on this. The mission profile is targeting 500 kilometer altitude. Just for reference, the International Space Station is orbiting around 429 kilometers, so we are going a little bit higher altitude. It's a polar orbit, still a low Earth orbit, but it's another trajectory. We are going to see more in details. And the mission duration is 3 orbits. It's going to be around 4.5 hours. And the sample retrieval is going to happen not sooner than August, because the capsule is going to be transported back to Germany after the splashdown. And also we need some time to make the samples and post them to our customers and to our partners. The Vandenberg launch site provides these different trajectories to be launched the rocket to. So there are differences compared to the equatorial ones, and we are going to see what are the main differences. But why we were happy for the opportunity to join them is because of this polar orbit. So astronauts were not in polar orbit until now, the recent SpaceX Dragon spacewalk, right? Before that was not in practice because of higher radiation and other reasons. So it's a very interesting orbit in which we can mimic longer space travels. So that's why we decided to go to this short mission, but this mission provides us to retrieve our samples. So as you can see here, we made a little summary about what are the different environmental conditions which we are going to face with. Basically the next demonstrator, the Mission Possible space capsule with the MayaSat-1 inside, they are going to cross these polar zones which are filled with discharged particles, and that's why it's very interesting compared to the other equatorial orbits which are not crossing this north or southern polar regions, which basically charged particles and different molecules are because of the Earth gravitational pull, it's coming down, which we are basically protected from, right? So we are going to, on purpose, cross these zones. So here we made a little summary about what is the main differences between a polar and the equatorial orbit. Just the key numbers. The radiation is 3 to 5 times higher, the TID in the polar orbit than equatorial. The charged particles, which I mentioned before, these zones, these flux zones, can be 10 times or even 100 times greater in the polar orbit. And obviously the cosmic ray radiation is 2 or 3 times higher than most of the equatorial or mid-inclinational LEO orbit. So with that, we are expecting changes on the molecular level, on the genomic level, or oligosaccharide level, or any indicators which are responsible for stress indication in these samples. In the plant's case, some of these modifications, let's call mutations, or these changes in the genomic level, are happening not in every specimen's case in the first generation. In some of the plants' case, we have to wait for the second and the third generation to observe any changes. And this provides a very good opportunity for a collaborative effort, and we can share knowledge and see what are those plants which are the viable candidates for longer term missions. So this is basically the mission objectives of the MayaSat, to select the most promising candidates for longer missions. And we try to choose the payload or research payload according to the space heritage. Namely, we prefer the payload which was not flown, which haven't flown before. So there are altogether 24 different customers on the MayaSat, representing 66 unique payloads, and altogether is hundreds of different specimens, algae, seed, and microbes, different microbes, and human DNA. So in this February 15, our customers and our partners are presented their research setups and their expected results on MayaSat. And the Genoplant Research Institute, we hosted the event in Budapest. So if you want to replay these presentations, there are very interesting algae research and soil microbiology research, or guests were Dr. Robert Zublin, the president of the Mars Society, and Dr. Zublin also delivered a speech on human missions to Mars and basically the long-term objectives. So it's worth to scan the code, and you can catch it after the presentation. And so our customers are going to be represented by Dr. Anna Sandak today. She is one of our earliest customers who decided to join us on MayaSat-1. And she's going to share her research insights. And it's a very interesting fungal biofilm project, which is a multiple scientific award winning project. So Dr. Anna Sandak, you are welcome on the stage, and now you can share your screen.
Okay, thank you, Bence, for the invitation. Okay, I will be sharing the screen and going for the full mode. Okay, I hope you can see my presentation. Yes. Yes, can you see it?
Yes, we can see.
Okay, great. So thank you again for the opportunity. And then yes, it's 1 month to go. I'm very excited and like looking forward. Then the mission will be successful, and I'm super happy that it's still possible. And then let's hope what the mission will bring to us. And then I will show you today very shortly what is our experiment about. But just to give you a bit of background, we are developing alternative coatings for the building sector. And when we think about the coatings, they are meant to protect the building materials and also to give the nice aesthetic appeal. And when you see the numbers here, there is a lot of billions of dollars like projected. And then, of course, the new buildings will be created in the future, and the new coatings will be needed. But however, when coatings are exposed to the external conditions, they are suffering the weathering process. And then this is like the combination of the biotics and abiotics factor. And as you can see here, the coatings often fail independent on the surface they're supposed to protect. And this is something which is happening. And then what is our human approach? We are trying to improve the coating performance, and very often we are adding the biocides or mineral oils or some synthetic chemicals, which maybe slightly improve the performance, but they are very environmentally unfriendly and toxic. As biologists and material scientists, I'm always thinking, okay, let's learn from someone who has more knowledge than us. And I'm truly fascinated by nature. And why nature, you will ask? And I think the answer is very, very short. The nature has 3.6 billion years of experience, and versus us humans on the earth, only 300,000, I think we can really learn a lot from the nature, how the nature is solving the processes. And materials in nature has very nice properties. This I will call them self-properties, such as self-growth, self-adaptability, self-assembling, or self-healing, as in our body, for example. And the question is, what if materials which this characteristic could be made? And what kind of new application will be possible if we will have these materials with the self-properties? And then the topic come here as engineering living materials. So this is something we are developing in our laboratories. Those are materials which are very unique because they are composite with the living cells. And then they are different categories of this engineering living materials, entirely bio-based or hybrid materials. But nevertheless, we are embedding living cells and we are keeping them alive. So the cells, different cells can provide new functionalities which are very hard to achieve for the conventional materials. And then if I will just summarize this in simple words, we are adding new dimension to materials which is not yet existing. We are adding life. And then our project is coming to the place. I'm working in the building sector for several years, and I'm offering witness of this not performing building the coatings, conventional coatings. And I was thinking that if we will be like looking how the nature is solving the problem, maybe we will be able to take the inspiration from the nature, and we will be able to create alternative technology for the protection of the materials. And here we are working on the ArchiSkin project, which is called the bio-inspired living skin for architecture. And we are trying to develop biofilm, so which is natural consortia of different microorganisms. Here we are working mainly with fungi, which has protective capacity. Our coatings are meant to work on different substrates because, as you know, in architecture, we are using not only concrete, but we are also using stone, steel, plastic, bricks, or wood. And then thanks to this living cells, they have several properties and functionalities which are very interesting for the building sector. Our candidates, which we selected after several trials, is Aerobasidium pullulans. Is the fungus, which is ubiquitous. It means it's living in different climatic zones. Is polyextremotolerant, so can survive very different temperatures and drought. So it is the fungus which you can find in the glacier, but also on the desert. So it's living in different environments. Has high phenotypic plasticity. It means then can switch the mode of growth from one cell type to another. And this is somehow allowing us to control the growth. Has also broad enzymatic profile. It means then we can feed the fungus with different nutrients, and those nutrients doesn't have to be very sophisticated and expensive, which is very relevant for the building sector, because whenever we think about the coatings are applied on the huge surfaces. Has broad pigmentation, so we can expect to have coatings with different colors, which is relevant for architecture. And finally, it's also not toxic. It's the biosafety class 1. So this is environmental fungus, which is present anyway and doesn't cause any problems for the humans. And of course, we don't want to have something which is problematic for our health. And then how can we boost the development of such materials, engineering living materials? And then what we do in the laboratory is this path: design, build, test, and learn. And of course, this is something which we are developing. But when I heard from Bence about this mission possible, we switched to another approach. What doesn't kill you makes you stronger. And then I was just thinking, what if we will be using this space condition as a booster for changes of our fungus? And maybe it will be much more robust for our application on Earth. So Bence was already showing the slides, but what we can expect is microgravity, higher radiation, limited nutrients, and oxidative stresses. All of these are harmful or problematic for the fungi to survive. But whenever it will be surviving, it will be stronger than the strain we were sending. So this is also something we will be investigating, and we are super interested if this is really working as we were thinking. So here, the mission possible. Again, we are very excited to be a part of this wonderful research initiative. And then, of course, in the experiments in space, there is like if you make mistake, is harmful because you cannot change it as easy as you do in the laboratory. So I was reading recently the paper about that, and the design of experiments can make the difference between the success and the failure. So I hope what we were preparing, it will be the success. But of course, this mission is very unique because the payload is coming back and nobody really knows what will happen and what will be the state of the samples we are sending. So this is making also this very, very exciting. So what is our experiment? We had this fungal biofilm that we are developing as a living coating for architecture. We selected strain which we are working with, and then we have we prepared the 5 payloads, and then we have also some control samples in our laboratory. But this is how we approach this experiment. So one, we are storing in our place in the 4 degrees in temperature. We have original strains, which is stored in -80. So this is our fungal collection. And then we have the payloads, which were like sending to Bence, and they are integrated with the capsules. And currently they are traveling to US for the launch. And then what we are planning to do, and of course, because we have the stored strains, we will be doing everything on the control samples and on all the samples which will be coming back. So we plan to have to check morphology with microscopy, with environmental scanning, electron microscopy, and fluorescence with some staining. We will do whole genome sequencings to detect mutation and any potential genetic differences. We will do NRI sequencing to analyze gene expression and compare the protein expression. Then we will be also testing the fungal growth and survival. So the fungi which will be coming, they will be already after some stresses, and we will try to evaluate if they are much more resistant than the control samples to the UV, oxygen doses, and temperatures. Then we will be also co-culturing the strains with pathogenic fungi to see if they are they have better inhibition, and then they are like much more robust. And finally, we will also analyze the metabolic changes. So what we expect as the outcome, we hope to have some differences in the biofilm formation and morphology. And then if we'll be able to observe phenotypic plasticity, that will be really great. Then maybe we'll observe some mutation which are linked with survival. And then fungi might become extremotolerance and then can have some metabolic shift. And then finally, we'll also observe epigenetic regulation of adaptation. And all of this outcome is, I really hope it will make engineered living materials much more resilient. And then having them back, we hope then coatings which will include such fungi which were like experience the space condition might be even more resilient to environmental change on Earth. So thank you so much for the time. I hope I was brief enough to present our project. If there are any questions, I will be happy to share. And
Thank you, Anna.
Yes.
Thank you so much. We are going to collect the questions in the chat box. So if somebody have any comments or questions, they can drop it there, and we are going to read you in the end of the presentation. Nobody sent questions yet, but we are going to wait. Maybe they will be want to know more. Thank you again, Anna, for sharing your research insights, and congratulations again for the Archiskin project, which was also partly supported by the European Research Council and is just amazing initiate. And we are also very pleased to be part of your journey. Anna, thank you. Okay, so back to our presentation. If you are interested to watch Dr. Anna Sandak full presentation, which was discussed in detail in the Genesis Space Bioscience Conference on February 15, you can scan this QR code, or you can find us in our YouTube channel, and you can find this playlist easily. It's worth to watch all of these presentations, because you can have a better understanding what our partners and what our customers are doing on MayaSat, and what was their reason they decided to go for this special orbit with us. So if you missed onboarding MayaSat, you don't have to worry. We're now working on a new initiative, which called Space Seeds Open Research Hub. It's going to be a platform very soon ready, in which you can log your space flown seed experiments. So there are a list of seeds which is part of the Genesis experimentations, and from these seeds, many now available for research or for commercial purposes, and basically anybody can reserve one of these seeds. On the platform which we are going to receive access in August when we are going to retrieve back our samples. You can log your experiment data, and so when you have an experiment of the chosen plants, you can log the seed type, also the observations, and you can involve also different soil simultans like Martian or lunar simultans, both going to be available soon in our web store. It's a very interesting thing, and if you want to be part of a bigger research and help us to support our database, we are going to publish your results. First, in an academic journal is Electrophoresis, which offering special issues for space research. I co-authored a couple of articles already in this journal. And basically, you have to involve some kind of electrophoresis or separation method. In this case, we can include you to this study if you want to be part of this publication series. The other thing is a book, The Genesis, The New Beginning, which is now available on Amazon. And in the second and the third editions and the continuous editions, we are going to always include the new results from our community, from our research hub. So just very briefly, I won't go through all of the list. We are going to make available 20 different seeds in this platform, which are already available, some of them on the platform. So I'm in our web store. In the okra seed, it's very important because it was not flown in space before, and it's interesting for those who are in the polysaccharide research. Me, myself, and my research group were more in the oligosaccharides part, so they are also sugar structures which are linked to different proteins. Either way, these sugars are very interesting indicators for many of the stress, abiotic and biotic stress indicators, which the protein supposed to suffer. And these sugars are going to be, these bonds, this link is it breaks, and it's going to go to the surrounding liquid. In the human case, this is the blood, right? And in the plant case, this is the different psyllium shot and all of the liquids which are going up and down the plant. So carrying the different nutrients. So from this liquid, we can detect these kind of sugars, from which we can conclude many interesting things on the protein stress. It's a fast growing crop, so post mission analysis, it will be very easy because you don't have to wait years for the results, and also rich in antioxidants, which is also very relevant to the space industry. We have a relatively limited total supply of these seeds. The rarity is rare, and obviously it's come with the certificates and the space heritage and the phytosanitary. The phytosanitary certificates, it's need basically for export if we are sending to overseas this. We have also a policy, so if our current customers, early customers, or the new customers who are now reserving seeds from us. If in case of any catastrophic event happened, we have a payload insurance. So we are going to repeat these experiments because Anna was also joking a little bit, but it's half joke, half serious. What if, I mean, obviously we are cheering for the successful of the mission possible, but in case of, for example, the rocket explodes or during the reentry, the capsule, burn down, we are going to repeat all of the MayaSat, and we are going to provide the same experimentations. But obviously, we are very positive, and we are going from the June launch, very good success and successful retrieval as well. So the deal seeds was in space already. So those who wants to have this seed, they have already a reference, a prior study that was aboard the Cosmos satellite. And these deal seeds are a fast germination and also have a very short life cycle. So you can have experimentation results very quickly. Why it's important to the researchers, one of the reasons is the antimicrobial compounds that this plant is producing. We have total supply more than 2,000 of these seeds because they are a small and light seed, so we could onboard many of these. Red cherry chili, they were not in space before. However, some of the variotypes like the capsules, like generally the capsule species was flown in space. Since 2021, there's a fruit crop study performed by the NASA on the International Space Station. Anyways, this variotype, the red cherry chili, was not in space before. At least there's no publicly available data, so this will be the first mission for that. We are going to have 200 seeds from this, and this is what we are very proud of, the Slovenian pumpkin seed. The pumpkins were one type of, not the Halloween pumpkin, but one of their cousins was in space before, but the Halloween pumpkin and the Slovenian pumpkin were not flown yet. So it's a very big seed. It's a very rare item. We onboarded only 40 of the Slovenian pumpkin, which are used here for their very rich oil. So if you are in any of this carotenoid research, it's also have antioxidant properties, and also can be very interesting to measure, especially the effect of the radiation, but also the effect of the microgravity. Tomato seeds. Tomato is the plant which we very like, I mean, in the plant science and also in agriculture, because it has the richest biological information among all the other plants and crops. For example, the first DNA map was published by tomato. It's very similar. It's like control plant. So when you are interested in discovering new markers or new structures, for example, when we were into the free and linked glycans, the first plant test was the tomato plant. So that was many tomato in space, especially since 2021, because of the Tomato Sphere initiated by NASA. They are involving many universities and students and primary schools also for tomato experiments. But this variotype, the Saint Pierre, was not haven't flown in space yet. So from this Saint Pierre, we are having a total of 800 supply. And this is the last one, the winter salad seed, which I'm going to present today. I'm just selected a couple of them, what was interesting for us for reasons, but we are going to upload each and every which is commercially available on our web store. Winter salad was not flown in space before, although the salads generally are very likely to study in space. The Waldman green and red leaf Lectuch was already in the ISS before. A very fast-growing cycle makes it an excellent candidate for these studies, and it's cold tolerant variotype. Rarity, we have 3,500, so it's not a rare item. This seeds is relatively small, so that's why we could onboard many of these seeds on the MayaSat. If you are interested any of these seeds on the list, you can reach them on the Genoplant web shop. We already uploaded a couple of those, but altogether, until the launch date, the total of 20 different seeds are going to be available. You can scan the QR code here in the top corner, or you can find the URL after the meeting, and you can map up the seeds. All of this data, what I shared here, like namely that was flown in space before, and why it's interesting for the scientific community, and the rarity data is also available in the product description. So if you are not sure which plant fits your study, you can have your study by the information, and after you can deepen this seek by searching on the concrete mission if that was flown in space before or not. The next missions, so if you want to join us for the next time, for the MayaSat-1 is too late, so it's fully booked and it's already handed over and already integrated, as you could see on the photos. Sometimes we're still receiving some requests if there's a chance to onboard MayaSat. No, unfortunately no. But you can have one of the flown seeds or biosamples which are available on our web store. This is how you can contribute. And if you decide to adopt one of these seeds, which you can see. I think the cheapest one is 2.9 euro per seed. So we are keeping the cost low in order to we can have many repetition and many different experimentations. You are going to contribute to support our database, and this is a very good thing to do because that's how we can make smarter conclusions. If not only one plant is tested in one laboratory for one test, but also you are performing even at home visual observations and basic measurements what you can do. Even though you can send your samples for post-mission analysis to the Genoplant laboratories, we are having a microchip electrophoresis device, so we can test for most of the total elements, micro and macro elements for saharids, for different compounds. You can also consult with our team if you are interested in one of the electrophoresis technique what we can do for your plant. So the next mission is going to be in 2026. We are building a GenoCube satellite. It's going to be a deployable one unit and with the purpose of testing multi-sample NDVI camera, which I'm going to present you very briefly. And it's a very new thing, the 2027. Now we are working on the funds, but soon we are going to share great news with you. We are going to build up a Gen. It's a 50 kilogram capacity capsule. It's a small scale re-entry vehicle, which we are going to perform the first re-entry test in 2027, and we also accepting early customers on this. It's going to be a retrieval mission, so it's going to perform a re-entry after the mission, very similar to the mission possible, what we are going to experience in the 2025 on the MayaSat-1. In the GenoCube case, we won't able to retrieve the samples, so it's going to be deployed by its own orbit after it was hosted on a larger satellite. But the experiment period can be up to 2 months, so it's a very promising platform, and I'm going to share with you what we are testing now. The 2027, it's going to be a short mission, but we are going to able to retrieve the samples, and you can perform post-mission analysis. The exploration company also planning to go in 2028 to the ISS with the bigger version of the NIX, which is going to be 3 tons capacity, if I'm not wrong. And we also are discussing to possibly contribute with a smaller payload with it's also a microgravity laboratory device, but it's a smaller payload. It's also going to be it's also going back to Earth after it was outside mounted to the ISS. So it's a very interesting thing if you are interested in any of these missions. So I just want to share with you what we are going to test in the 2026. So in this one unit CubeSat, it's an NDVI camera is going to be tested, which you should know that this kind of sensors are basically used for mapping a chlorophyll index from vegetation, and basically they are linked to larger satellite, is a satellite, not satellite, mapping up larger forests and larger areas. In the GenoCube case, we are going to apply this camera for interior observations, and we are now working on an algorithm in which we can use the same camera to read out multiple samples at the same time, which was not common in the practice before. So what you can see here is different samples, starting with the control sample in the middle with the lowest number of algae cells, and we are increasing the concentration of the algae in this solution. And you can see by one camera we are able to detect even small differences of chlorophyll. So this is the 2026 mission about. And just very briefly about the Gem capsule. So it's going to be a small scale reentry vehicle with a maximum launch payload mass of 50 kilogram. And we are going to test a heat shield for one of the customers. And it's also going to be part of ESA RPA application. And so we are very happy to the team is now putting together the gen, because with that we are increasing our capacity for re-entry and we are going to be more independent. So we, after a while, when it became operational, we can have more frequent launches, even more than once a year. So if you are interested to fly with us in one of our missions, which is coming, scan this QR code, or you can find our contact information in the Genoplant, and you can set up a meeting with the team. I would like to thank you very much for your attention, and if you have any questions, please don't hesitate to share with us. And we are going to read if we received anything so far in the comments. Okay, no specific questions. Thank you very much again. And we are looking forward to hear from you if you need samples. We received a question from Terry. I wanted to grab a few radish seeds and the lactage plus the patch, and it would be keen to fly with you as well. But I tried to check out in your store, and the shipping cost is over €1,000. Can you clarify this content? Yes, it must be something wrong with the shipping. So I'm going to check the WooCommerce. Maybe something went wrong. It shouldn't be €1,000 for shipping, obviously. I'm going to help with you. Can you please drop your? I'm going to send you a DM, Terry.
I dropped my email address in there.
Thank you so much. I'm also just wanted to drop you mine in the comments. So yeah, we are going to help you out. Thank you very much.
Very exciting. Great work.
Thank you. The platform is very new, so we just started to upload the seeds, and there's very possible there's some mistakes yet. Thank you so much for warning us.
Copy that. No worries.
Well, thank you everyone for your attention. You can also reach us on LinkedIn, and we are going. That was the last meeting before the launch event. So if you want to participate in California, near to the Vandenberg launch pad, or you want to celebrate with the European team in Planegg, Germany, please also let us know, and see you soon in June. Thank you. Bye-bye.
Thank you.